Sheep and goats are valuable livestock species that have been raised for their production of meat, milk, fiber, and other byproducts. Due to their suitable size, short gestation period, and abundant secretion of milk, sheep and goats have become important model animals in agricultural, pharmaceutical, and biomedical research. Genome engineering has been widely applied to sheep and goat research.
Hi, Assalamualaikum. Now I want to present to you about the history of method. First, the method is early genetic engineering techniques. In 1972, Paul Berg and Phyllis developed the first recombinant DNA molecule by combining DNA from different sources. In 1973, Stanley Cohen and Herbert Bayer successfully transferred a recombinant DNA molecule into bacteria, established the foundation for genetic engineering. Second, transgenic technology. In 1980, the first transgenic mouse was treated by injecting foreign DNA into a mouse embryo, marking a significant step towards genome manipulation in animals. In 1982, the first transgenic plant, a tobacco plant resistant to antibiotics, was generated, the zinc finger nucleus. In 1996, scientists discovered that zinc finger proteins could be engineered to bind to specific DNA sequences, leading to the development of zinc finger nucleus as a gene editing tool. Zinc finger nucleus use a DNA targeting enzyme called POC attached to zinc finger proteins to target and induce double-strand breaks in DNA at desired location.
There are some applications of genome engineering. Firstly, genome engineering can be used to enhance agriculture plants, making them more resistant to pest, disease, or environmental challenges. Second, genome engineering holds great promise for treating genetic disorders and disease. By modifying specific genes, it's possible for disease. Research can politically correct genetic defects and restore normal function. Next, genome engineering has industrial application in a variety of fields, including pharmaceutical, biofuel, and bioremediation. Genome engineering can be used to improve livestock breeding by adding desired features such as disease resistance, increased growth rate, or higher meat quality. Genome engineering can help with environmental conservation by addressing risks to ecosystem and endangered species. Lastly, genome engineering also can present significant biosecurity and ethical questions.
Advantages of genome engineering. Firstly, disease treatment and prevention. Genome engineering offers the potential to treat and prevent genetic disease by correcting or modifying specific genes associated with these conditions. Next is agricultural advancement. Genome engineering has the potential to improve crop yield, enhance nutritional content, and increase resistance to pests, disease, and environmental stress. Third is drug development and biotechnology. Genome engineering techniques can be used to develop more effective and targeted drugs. Next is basic scientific research. Genome engineering allows researchers to study gene functions and their roles in biological process more precisely. And lastly, conservation and environmental protection. Genome engineering techniques have the potential to aid conservation efforts by protecting endangered species and restoring damaged ecosystems.
Disadvantage genome engineering. The manipulation of an organism's genetic material, known as genome engineering, has the potential to revolution industries such as medicine, agriculture, and biotechnology. However, as with any strong technology, it has drawback and raise ethical consideration. The following are some of the drawbacks of genome engineering. Off-target effect. Genomic engineering tools such as CRISPR Cas9 can sometimes result in unwanted genome charge. Deaths of target impact can introduce new genetic variation with unanticipated consequences, such as potential injury to the organism or independent charge to other genes. Ethical concern. Genome engineering creates a number of ethical issues. For example, the technology might be used to change human embryos, raising concern about the development of designed baby and the possibility of eugenesis. Furthermore, the use of genome editing in non-human characters, such as the correction of genetically modified crop of animal, raises worries about the environmental impact or unforeseen repercussions for ecosystem. Incomplete comprehension. Despite considerably advanced in our understanding of genetics, there is still much we don't know about the genome complicated. Modifying genome with a thorough understanding of their activities and interaction might result in unforeseen consequences as unpredictable long-term repercussions. Long-term impact are unknown. The long-term implications of genome engineering are not entirely understood. Modifying an organism genome can have anticipated implications that emerges over time or in future generations. To mitigate potential risks, substantial study and rigorous safety assessment are required. The rapid evolution of genome engineering technology has overtaken the direction of relevant legislation and norms. This threats difficulty in assuring responsible and safe technology use, as well as addressing issues about accessibility, equal distribution, and potential misuse. The availability and accessibility of genome engineering technologies may result in socio-economic inequalities. If these technologies become predominantly available to a privileged few, current inequalities in healthcare, agriculture, and other sectors may worsen. It is critical to highlight that while genome engineering has drawbacks, it also has tremendous potential benefit. Responsible research, stringent safety measures, and detailed ethical consideration are required to maximize the benefit of genome engineering while minimizing the risk.
The most important method used in sheep and goat genome engineering is embryonic cell cloning. Embryonic cell cloning is mammal can be intentionally cloned utilizing methods like nuclear transfer and embryo splitting. Nuclear transfer is a more sophisticated approach that enables the generation of cloned progeny from adult cells. Transgenic animals with desired features have been created using cloning. Cloning is a contentious topic, but it could be a useful tool for science and medicine.
The following techniques can be used to increase the effectiveness and rate of embryonic cell cloning in sheep and goat genomes. The first one is selection of donor cells. Choosing the right donor cells is essential for a successful cloning process. Embryonic stem cells or early-stage embryonic cells, which have a high rate of proliferation and little differentiation, are preferred. These cells can help a healthy clone embryo grow since they have a greater capability for reprogramming. Number 2 is synchronization of donor cell and recipient oocyte status. The success of cloning can be increased by coordinating the development phases of the donor cells and recipient oocytes. To ensure compatibility and promote successful fusion and development, it is crucial to collect oocytes and donor cells at the right stages. Number 3, optimization of culture media and conditions. When it comes to promoting the growth and development of cloned embryos, the culture material and circumstances used throughout the cloning procedure are extremely important. The effectiveness of embryonic development can be increased by carefully adjusting the culture media's nutrition, growth factor, and hormone concentrations. Number 4 is nuclear transfer technique. The success of cloning can be affected by the nuclear transfer procedure, which includes inserting the donor nucleus into an uncreated oocyte. Enhancing the nuclear transfer process can increase the effectiveness of cloning by optimizing fusion settings, applying cutting edges and micro-manipulation methods, and timing fusion. Lastly, epigenetic modification. The formation of donor cells nuclei after reprogramming by epigenetic alteration is important.
Next, I will present about discussion. Since the 1970s, tremendous progress has been made in sheep and goat genome engineering. The early technique had a high off-target impact rate and were wasteful, such as random transgenesis. In more recent time, genome editing has become more accurate and effective because of the creation of site-specific nucleus like CRISPR. In order to better understand gene function, enhance animal breeding, produce medicine in meat, increase animal illness resistance, recapitulate human disease, and serve as hosts for the development of human organs, genes-edited sheep and goats have been developed. Pharmaceutical manufacturing in meat is the one of the most exciting use of gene editing in sheep and goats. This strategy may offer a reliable and long-lasting supply of therapeutic protein for human use. One such instance in this genetic modification of sheep and goats to produce human antithrobin, a protein that assists in the prevention of blood clots. An unusual disorder called congenital antithrobin deficiency can result in life-threatening blood clots. This treatment may benefit people with this condition. The enhancement of animal breeding is promising new use for gene editing in sheep and goats. For example, genetic alteration has been employed to increase the muscular mass of sheep. Additionally, the quantity and quality of meat suitable for human consumption may increase as a result of goats' capacity to produce more milk as a result of genetic mutation. Lastly, gene editing has the potential to transform the way we think about cattle production in addition to this particular use. By providing us with the capacity to precisely alter the genomes of sheep and goats, we can generate creatures that are better suited to our needs. As the consequence, new products and services that benefit both people and animals might be produced, and livestock could be produced in more efficient and ecologically responsible manners. Genome engineering in sheep and goats has a promising future. We may anticipate seeing even more cutting-edge use for the portion instrument as technology advances.
Next, I will share about step on embryonic cell cloning. For the first step is selection of donor cells. From a donor goat or sheep, remove somatic cell, which is non-reproductive cell. Secondly, inoculation. An unused egg taken from a female sheep or goat should have its nucleus removed. Next point is cell fusion. Use an electric pulse using a thin glass needle to introduce a somatic cell into the unplated egg to promote fusion. Next point is activation of the reconstructed embryo. Use a particular culture medium or an electric pulse to stimulate the restored embryo to begin the development. Next is embryo culture. In a culture dish with a suitable growth medium, place the activate embryo for development. Next point is transfer to surrogate mother. Glycosis transfer into the ovaries of a sheep or goat used as a surrogate mother. For the last point is pregnancy and birth. A clone sheep or goat is born after the clone embryo was carried by surrogate mother to term.